First of all, I would like to thank the organizers to give me the chance to present my recent work. This work has been done by me and my supervisor, Guido Sanguinetti. Now I'm going to introduce a methodology that can do the splicing quantification in single cells. First, let's look at what is the RNA splicing and what is the challenge in splicing quantification in single cells, and whether there is any possibility to solve the challenge. So RNA splicing is a biological process where the long coding region will be cut off and the coding region will be joined together during the transcription and before moving to the translation. And we call the long coding region as introns and the coding region as exons. In many eukaryotic genes, there are multiple exons. So one gene can produce multiple transcripts just by different combinations of the exons. For example, in this gene, there can be two different transcripts. The first one is the combination between exon 1 and exon 3. And the second one is the exon 1, exon 2, and exon 3. And we call it the different isoforms. And this process calls autotative splicing. Autotative splicing is very important, not only because it largely expands the complexity of the proteomics. For example, 20,000 human genes can produce 200,000 proteins, roughly. And more importantly, it's a key regulator of many biological processes. For example, in this case, if the DNA got damaged, then a specific isoform is required to help the cell survive. So, how to quantify the splicing? Or given a gene that have multiple isoforms, how to quantify the fractions of each isoform? One of the most powerful methods is RNA-seq, as all of you know, and however, the measurement is not that direct because of the length of the reads is usually short. It's much shorter than the transcripts, and in most cases, it's shorter than the exon itself. So you will see a lot of reads mapping to the shared exon, for example, exon 1. This read mapping to this exon, and then you lose the direction information of the identity of these reads. It may come from the first isoform, or it may come from the second isoform. In some cases, you may see that there are some reads mapping to the exon-exon junctions. So you will see two exon-exon junctions here and three exon-exon junctions here. Then you could give a direct estimate of the ratio between the two isoforms as 2 to 3. However, the problem is that you waste a lot of reads. Then people suggest that why not use a probabilistic distribution and then use the psi as a parameter of the distribution. And one of the most popular model is the mixture of gapped uniform distribution. So the likelihood given the psi is a fraction of all the isoforms. And the likelihood is like this. It's a mixture model, and it's a product of all the rates you observe. And unsurprisingly, this model works very well. The framework has been first introduced in the meso model, which is a few years ago, and they showed that in different coverage, which is the rates per kilobase, that in different coverage, the estimates variance in the probabilistic model is much smaller than the direct methods, although both of them are unbiased because it's the simulation and the truth is 0.5. So this problem is almost solved in bulk RNA-seq in terms of the accuracy and the computation speed. For example, recent years there are like Carnisto and Firefish or other more methods. But how about the single-cell RNA-seq? The challenge is the coverage is much lower than 250. It's like very far from here, like 50 or even lower or even no rates. So let me quick remind you the technology noise in single-cell RNA-seq. The correlation is low because of either the technical noise or the high dropout. So you will see that for many genes, although it's expressed, but you can't see any rates because a dropout happens. So when you look at the example, there are 6 cells for this DNMT3B genes, which is the key regulator of DNA methylation, and the splicing happens on these exons. So the density of the sashimi plot is the coverage. How many rates are here? And the later numbers are the junction rates. So for this cell, the including or exclusion for this exon is like a half-half. And for this one, it's like a all exclude. And for some other cells, the coverage is very low. And for many more cells, there is no RNA-seq rate at all. Maybe because of the low expression, or because of the dropout, or because of the very low coverage sequencing. Different from the gene expression level, the challenge in splicing is the very low coverage for many genes, as there is no like challenge for normalization or other else. Because of the very low coverage, essentially you don't have data, then people even suggest trying to avoid the splicing analysis 2 years ago. And but from the machine learning angle, you may want to see whether we could do the imputation or do the prediction if we don't have the data or we don't have sufficient data. Actually, a few years ago, people already see that the splicing is highly predicted from the surrounding sequence. For example, here the Brandon Frey's lab produced the splicing code, which is over 1,000 sequence features, and they use a neural network model to predict the splicing, and it can be predicted very accurately. We tried this prediction. We used only the sequence feature around the splicing, and we use a simple linear regression, Bayesian linear regression, and we can already get a good correlation between the observed splicing and the predicted splicing. This is by cross-validation rather than on the training data. So this gives us an idea is that why not combine the prediction and the quantification together? So we propose a method called Bayesian regression for isoform estimate, BRIE for short. So in these methods, if you have sufficient data, then it goes to the quantification. This lets the data speak loudly and is consistent with the estimate without the prior. If there's no data, then it goes to the imputation by the sequence automatically prediction. If it's intermediate, then it goes to the posterior combined by the prior and the likelihood. as we focus on the posterior distribution, here is the graphical representation which shows the dependency of these variables. The variables in shade is the observed variable, for example, the rate, the location of the rate, and the sequence features. And others are latent variables which we want to infer. And the left half is a Bayesian regression, so you can use a Gaussian distribution for the Bayesian regression. And the inner model is the mixture model that, given the psi, it's the mixture model to observe the assert of the sequence rates. So from the graphical model, we could write down the full distribution, the posterior distribution, as like this. It's a product of capital K splicing events. This can be like 10,000. And for each splicing event, all of the rates at the mixture model. Here, as we focus on the psi, so we could collapse the weights analytically because it follows the Gaussian distribution in the Bayesian regression. Still, even we collapse the weights, they still have like 10,000 dimensions for the joint distribution. So it's very hard to compute, especially when we want to get the full distribution by MCMC sampling. So we use a heuristic way that, given the weights, we sample the psi for each splicing event independently, and then we update the weights again, and use the weights into the psi sampling again. In some cases, we could even fix the weights. For example, we learn the weights from the bulk RNA-seq or pulling the single-cell RNA-seq. Then let's look at the performance of our methods. First, we did a simulation from a bulk RNA-seq simulator because currently there is no very good single-cell RNA-seq simulator. So we just make the coverage as low as the RPK as 25. So we compare the BRIE with the informative prior or uninformative prior. As can be seen that with an uninformative prior, the correlation between the truth and the estimates is just over 0.7. But if we include a informative prior, then the correlation goes to 0.9. We then move to other coverages, and we see that BRIE with the informative prior always gives a better estimate, especially when the coverage is low. And very interestingly, we also see that BRIE with an uninformative prior also gives a very competitive performance at other methods like Canisto, MISO, and DISE-seq. That means if you don't have a good feature set, BRIE won't hurt the estimates. Then let's look at the performance in a benchmarking single-cell RNA-seq. The problem for the assessment is that we don't have the truth for the splicing in single cell, so we use the bulk RNA-seq as a reference from the same tissue. So you will see that with the uninformative prior, you will see many genes have no data, so the estimate goes to around 0.5. But if you give us an informative prior learned from the sequence feature, then the correlation goes up to over 0.8. Then we look at the more cells and more other methods, and we also see the similar pattern. Secondly, we look at the correlation between two single cells. Unsurprisingly, that many genes also centers to 0.5 in both cells because dropout happens or just low coverage. And by use the informative prior, the correlation goes up largely. And also for other methods. And then we look at the confidence of the estimate. Here we see that BRIE gives a more confident estimate so that there are more genes past the 95 confidence interval lower than 0.3. That means this gene can be used for downstream analysis. Now, BRIE can give the distribution estimate for the posterior as the black histogram. And also the prior distribution, if you can see, is the blue curve. So the other question is that whether we can detect the differential splicing. As we use the Bayesian method, then we also propose a Bayes factor to detect the differential splicing. Here we assume that the two cells have the delta as the difference of the two cells in splicing ratio, and the non-hypothesis is that there's no difference, and the alternative hypothesis is there is a differential splicing, and the data is the observed posterior distribution that we use the MCMC sampling. So the Bayes factor supporting the differential splicing is that equivalent to see the no difference just by the prior, but versus the probability to see the no difference from the posterior. So this can be done by resampling the posterior distribution and the prior distribution. So here is the result of detecting differential splicing. We compare the BRIE with other two methods, MISO, which also use the Bayes factor, and RMS use a false discovery rate. And by different thresholds, like a Bayes factor greater than 50, greater than 10, or greater than 3, we always see that BRIE gives a much higher sensitivity in detecting the differential splicing benchmarking dataset. However, even by BRIE, there are only between 1 to 2% of splicing events can be detected as a differential splicing, and we believe this is far from the false positives. And we also see that by another dataset on mouse embryonic cells that in the late stage, there are more differential splicing happens than the earlier one. So I would like to summarize here is that our methods BRIE as a Bayesian methods allows you to do the imputation if you don't have data, and also allows you to do quantification if you have a lot of data, or gives you a trade-off between them. And this framework can be applied to many other similar problems. And we developed the methods as a standard Python package. And here is the manual that I developed. And our paper has just come out a few weeks ago in Genome Biology. And if you want to look more details, you're welcome to download. And finally, I would like to thank my supervisor, Guido Sanguinetti, gives me a lot of help during my PhD in the past 3 years. And also Jim Brooks, my second supervisor, who works on splicing for over 20 years. And Chris Ponting gives me a lot of suggestions on single cell stuff. And also the scholarship from a university for support. And all the people from Guido's lab gives me a lot of day-to-day help, yes. And especially Andreas and Anina are around here.
So I've two questions. The first is, I'm not a machine learning guy, but I am an RNA-seq splicing guy. It seems like your model has to learn from the data what the patterns of splicing are. If you perturb that with different conditions where suddenly the gene expression patterns change radically, like different stages of cell development, do you not have to relearn the model? So it seems like if you have strong differences between your cell types and their gene expression cell types, you really shouldn't be using the same learned framework for each in order to predict the splicing.
I got your question. Thanks. It's a good question. In splicing quantification, we said it's a self-normalized problem. For example, you may see that two genes have very different gene expression, while the splicing is similar, or the splicing is different. Then you cannot, it's very hard to tell whether the gene level is the main regulator or the splicing level is the main regulator. I think it's a very hard problem and I haven't tried to solve it. Thanks.
I'm nervous about imputing data on conditions if my model's been taught on a wild type or something like that. I'd be nervous about that. And the second point I wanted to make is you made some comparisons with the other tools, and you mentioned Callisto and sailfish and salmon, but obviously they're actually tackling a slightly different problem. So they're not defining a transcript just through splicing, because you can have two transcripts that both have a single exon, both have exactly the same exon and have different UTRs, which are also important in expression. And obviously your tool only deals with splicing at the moment. So do you have any plans to take that to look at whole transcripts and transcript quantification rather than just splicing quantification?
Yes. When using the Callisto or sailfish or other methods, which based on the transcriptome, so I generate the transcriptome by myself from the annotation. So I would assume that the annotation, no matter by the GTF file, will just use the transcriptome sequence file. I assume that they are the same.
Okay, well, we'll talk about it.
One last quick question and a quick answer.
Right. So I saw in the comparisons your genes tended to default to 0.5 without informative prior. Is that correct? So does that mean you only use 2 isoforms per gene in your? Was that a simulation?
In all the work, because if you look at many isoforms, it's very hard to give a good prior.
Right. Yeah, because many genes have more than 2 isoforms.
Yeah, so we look at the simplest thing.
Right. Okay.
Yeah.
Thank you.